Introduction to Module 2. In our first module, we explored how to control microbial growth, prepare and maintain cultures, and work with pure microbial isolates. In this second module, we will shift our focus toward observing, identifying, and analyzing microorganisms. We will begin with microscopy, the cornerstone of microbiology. We will examine the principles of different microscopy techniques, learn how specimens are prepared and stained, and then move on to specialized analytical tools like spectrophotometry, fluorimetry, flame photometry, atomic absorption spectrophotometry, and chromatography. The goal here is simple. By the end of this module, you should feel confident not only in growing microorganisms but also in visualizing them, differentiating them, and analyzing their biochemical properties. Section 1. Principles of microscopy. Microscopy is essential in microbiology because microorganisms are too small to be seen with the naked eye. The principle is straightforward. A microscope uses lenses to magnify small objects so that they can be studied in detail. 1. Light microscopy. This is the most common form of microscopy in microbiology laboratories. Light microscopes work by passing visible light through or reflecting it off the specimen. The image is then magnified using a series of glass lenses. The resolution, the ability to distinguish 2 points as separate, depends on the wavelength of light and the quality of the lens. Types of light microscopy. Bright field microscopy. The simplest form, where light passes directly through the specimen. Often requires staining to see details because most microbes are transparent. Dark field microscopy. Uses a special condenser to scatter light so that the specimen appears bright against a dark background. Useful for viewing live, unstained organisms and delicate structures like spirochetes. Phase contrast microscopy. Enhances contrast in transparent specimens without staining. Works by converting differences in refractive index into differences in light intensity. Useful for studying living cells and observing motility. Fluorescence microscopy. Uses fluorescent dyes that emit light when excited by ultraviolet light. Very useful for detecting specific structures or organisms using fluorescently labeled antibodies. Confocal microscopy. Uses laser light to produce high-resolution, three-dimensional images of specimens, often combined with fluorescence. 2. Electron microscopy. Electron microscopes use a beam of electrons instead of light, which allows for far higher resolution. Transmission electron microscope, TM. Passes electrons through ultra-thin sections of the specimen. Reveals fine internal structures. Scanning electron microscope, SEM. Scans the surface of the specimen with electrons, producing detailed three-dimensional images of the exterior. 3. Specimen preparation for microscopy. To visualize microorganisms under a microscope, they must often be prepared using specific techniques. Fixation. Preserves cell structure and immobilizes organisms. Smear preparation. A thin film of microbial suspension is spread on a slide and allowed to dry. Staining. Enhances contrast between the organism and the background. Section 2. Staining techniques. Staining makes microorganisms more visible by increasing contrast and highlighting specific structures. 1. Simple staining. Uses a single dye such as methylene blue or crystal violet. Highlights the entire organism, showing shape, size, and arrangement. 2. Differential staining. These techniques distinguish between different types of bacteria based on their staining properties. Gram staining. Separates bacteria into gram-positive, purple, and gram-negative, pink, based on cell wall structure. Gram-positive bacteria have thick peptidoglycan layers, retaining the primary dye. Gram-negative bacteria have thinner layers and lose the dye during the decolonizing step. Acid-fast staining. Identifies bacteria with waxy cell walls, such as Mycobacterium. These retain the primary dye even after acid-alcohol treatment. 3. Special stains. These are used to visualize specific structures. Capsule stain. Uses negative staining to visualize the polysaccharide capsule. Flagella stain. Makes flagella visible under the light microscope. Endospore stain. Stains tough, dormant bacterial spores. Nuclear stain. Highlights genetic material within cells. Section 3. Advanced optical tools, micrometry and microdensitometry. Micrometry. Measurement of microorganisms using a calibrated ocular micrometer. Microdensitometry measures optical density in microscopic structures, useful in quantifying stained material. Section 4. Analytical tools in microbiology. Beyond microscopy, microbiologists use instruments to measure and analyze chemical and physical properties of microbial samples. 1. Colorimetry and spectrophotometry. Colorimetry measures the concentration of colored compounds in a solution based on light absorption. Spectrophotometry measures the amount of light absorbed by a solution at specific wavelengths. Used to estimate microbial growth by measuring optical density, turbidity. Can detect specific compounds produced by microbes. 2. Fluorimetry measures the intensity of fluorescence emitted by a sample after excitation with light of a specific wavelength. Often used for detecting minute amounts of compounds in microbial metabolism studies. 3. Flame photometry measures the concentration of certain metal ions based on the color they emit when introduced into a flame. Used to analyze sodium, potassium, and calcium in microbial culture media. 4. Atomic absorption spectrophotometry detects specific metal ions by measuring the absorption of light after atomizing the sample. Provides precise quantitative results. Section 5. Chromatography techniques. Chromatography separates components of a mixture based on differences in how they interact with a stationary phase and a mobile phase. Types of chromatography used in microbiology. Paper chromatography uses paper as the stationary phase. Often used for amino acid separation. Thin layer chromatography, TLC. Similar to paper chromatography but uses a thin layer of silica gel or alumina. Column chromatography uses a packed column to separate compounds. Can be adapted into many forms: gel filtration, ion exchange, and affinity chromatography. Partition chromatography based on differences in solubility between two liquid phases. Gel filtration chromatography separates molecules by size. Ion exchange chromatography separates based on charge differences. Affinity chromatography uses specific binding between molecules, like enzyme-substrate or antigen-antibody interactions. Gas-liquid chromatography, GLC, separates volatile compounds using a gas as the mobile phase. High-performance liquid chromatography, HPLC, uses high pressure to pass liquids through a tightly packed column, achieving high-resolution separation. In this module, we cover the essential tools and techniques for observing, identifying, and analyzing microorganisms. From the basics of light and electron microscopy to the specialized staining methods and advanced analytical tools like spectrophotometry and chromatography, you now have a comprehensive understanding of the ways microbiologists study the microbial world. When we combine the control and culture methods from Module 1 with the observational and analytical techniques from Module 2, we have the complete toolkit of a microbiologist.